Nutrition, Bhavan'sVC College, and has 6 years of teaching experience. She has a very good research expertise and has complete 2 postdoctoral fellowships. Coming to her educational background, she has completed her PhD from University of Kerala in 2010 and has been impacted gold medalist for holding first rank in both her BSc and MSc for University of Kerala. She has receiving JRF fellowships from CSIR in 2006, has been awarded Young Scientist Award 2015 and receiving Young Researcher Award 2024 for Sakambari Trust. She is skilled in biochemical aspects like protein chemistry, protein purification, cell biology, molecular biology, cell cultures, cell signaling, molecular genetics. And now I request Dr. Manju Devi to take over the session.
Thank you, Kamala ma'am. Am I audible?
Yes, you are audible.
So good evening, all the participants, respected Dr. S. Padma ma'am, Head Department of Biochemistry and Nutrition, and the faculty members, Dr. Vanitha ma'am, Dr. Kamala ma'am, and all dear participants. Welcome to the 5th day of the evaluated course. So let me start the session. I'm sharing the screen. My screen is sharing, ma'am.
Yes, ma'am. You can continue.
So, 1 second. So today I'm going to give you a presentation on the RNA. Okay, so today's topic is structure. Actually, the screen I want to share another one. 1 second. Now the screen is sharing, na ma'am.
Yes, Manju.
So the today's topic is RNA structure prediction tools. So up to yesterday, that is the last 4 days. Last 4 days, the resource persons were discussing with you about the various genomic tools, like how to retrieve the gene structures, how to align the gene structures, and what is the, how to predict the exon and intron of the gene sequences, and how to design a primer for the genes. So the functional aspects as well as the structural aspects of the genes, which is coming under the genomics. Yes, ma'am.
Screen is sharing. Maybe you can put it on the slide mode, the slides.
Yeah, sure.
Manju ma'am, full screen lo pettuko.
Now it is full screen?
Yes.
Yes.
Okay. So various aspects of the genomics have been discussed up to yesterday. So today I am going to focus on the transcriptomics. So what is meant by transcriptomics? It is the comprehensive study of an organism's transcriptome, which is the complete set of all RNA molecules, which includes the various RNAs such as mRNA, rRNA, tRNA, and other non-coding RNAs, which are produced within a cell or tissue or an organism at a given time. Okay, so today's topics will be about the various RNAs and how to predict the structure of the RNAs. Okay, so which will be covering under the transcriptomics. So what is the significance of studying the transcriptomics, which helps to understand the gene regulation process? It helps to understand how genes are turned on and off in response to various stimuli such as disease, environmental changes, or developmental stages. And studying of this transcriptomics will also help you to identify the diseases for the diagnosis as well as the prognosis of the diseases by analyzing the various gene expression patterns. Thereby, you can identify the biomarkers for the diseases and also can predict the disease progression and thereby personalize the treatment options. And transcriptomics will also help you for identifying and discovering the drugs. It can help identify new drug targets and assess the efficacy of the existing drugs. In agriculture also, the transcriptomics have an application. It helps to improve crop yields, enhance the stress tolerance, and develop disease-resistant plants. So these are the various applications and thereby significance of understanding about the RNAs and the transcriptomics. So now let us move on to the next slide. So as per molecular biology, the central dogma, what it states that it is the process where the genetic information flows from DNA to RNA and then from RNA to the protein. So today's our focus is on the RNA, which is mainly the process through the transcription, the RNA is synthesized. So what are RNAs? I want at least some of the participants to unmute and tell me what you know about RNAs. Participants, please, are you all there?
Yes,
what do you know about RNA? Already my slide also it is visible, but then what is your understanding about RNA? And because majority of the participants are UG students, I want to give an overview of the RNA before going into the structure prediction. So please respond. What do you know about RNA?
Has 4 bases. RNA, that is in the uracil.
So what is the difference between a DNA and RNA? Any idea?
Uracil. UNT.
In case of RNA, it is having uracil as the nitrogenous base, whereas in DNA, thymine is the nitrogenous base. Okay. And further, students, here you can see as it is shown in the picture, DNA is usually double helical stranded, whereas RNA is usually single stranded. Okay. And the sugar also different. In case of RNA, ribose is the sugar, whereas in DNA, deoxyribose is the sugar. And the nitrogenous bases in RNA are adenine, guanine as the purines, and uracil and cytosine as the pyrimidines. Okay. And we know that in case of RNA, it is a nucleic acid, and generally we believe that enzymes are only proteins. Okay. But later on, research have shown that even RNAs also have, some of the RNAs also have a enzymatic activity. So those RNAs which have a catalytic activities, they are called as the ribozymes. Okay. So in addition to their genetical role, even RNAs are functioning as enzymes also. Okay. So this is the basic structure of the RNA molecule, which is a single stranded structure. Okay. But we will be predicting the secondary structure of the RNA after giving an overview of the various RNAs. Okay. So how the RNA is synthesized, as the central dogma states, it is through the process of the transcription, where the two double helical strand of the DNA will be separated, forming one of the strand will be acting as a template for the synthesis of the mRNA, and the enzymes RNA DNA dependent RNA polymerase enzyme will be involved in the process of the RNA synthesis. Okay, so the DNA unwinds and one strand will be acting as a template for the synthesis of the RNA. Okay, and depending on the RNA, whether it is a mRNA or rRNA or tRNA, various RNA polymerases are there which will be catalyzing the transcription process and thereby synthesizing the RNA. Okay, and as all of you know, the major 3 types of RNA molecules which are present in the cell are the messenger RNA or mRNA, ribosomal RNA or rRNA, and transfer RNA or tRNA. Okay, so the mRNA, as the name name implies, mRNA carries the message of DNA for the synthesis of the proteins, okay? So, and that information is stored in the mRNA in the form of the codons, okay? So, the information will be genetic information will be flowing from DNA to mRNA, and then from mRNA to the functional product of this genetic information is the proteins only, and the mediator between the DNA and the proteins are the RNA, that is the mRNA. That is why it has got the name messenger RNA, okay? And in the mRNA, it is in the form of the triplet of nucleotides called as the codons, okay? And the second class of the RNAs which are abundant in the cell are the ribosomal RNA, which are also part of the protein synthesizing machinery ribosome. The ribosomes, where the synthesis of protein happens, it comprises of both the ribosomal RNA as well as the proteins. The third group of RNAs are the transfer RNAs, which carries the amino acids in the form of amino acyl tRNA for the corresponding codons in the mRNA. Okay, so these are the three major RNAs which are present in the cell. So here you can see the three RNAs. So now let us see little detail about each of these RNAs. Okay, so the mRNA, which is synthesized from the DNA. By the process of the transcription. Here you can see from the gene, the first a pre-mRNA is synthesized, which contains both the exons as well as introns. Okay. And do we remember what were exons and introns, participants? What is the difference between exon and intron?
Exon is a coding sequence and introns is a non-coding sequence.
Yeah, so exons are the coding sequences, whereas the introns are the non-coding sequences. So initially, mRNA is synthesized in a pre-mRNA form, where both the coding sequence as exons and non-coding sequence as introns are present. Then the RNA will undergo a post-transcriptional modification, thereby the introns, that is intervening, these non-coding sequences are removed by a process called splicing, okay, thereby producing a mature functional mRNA, okay. So after the synthesis of the RNA, it has to undergo a post-transcriptional modification, thereby removing the intervening non-coding sequences, that is the introns are removed, thereby producing a functional mRNA. In addition to this removal of the introns, during the post-transcriptional modification, mRNAs will also undergo some modification. Thereby, at the 5' site, a cap will be added, and at the 3' site, a poly A tail will be added. Okay, these are another two modifications which happens to the mRNA during the post-transcriptional modification. Okay, so this is how the mRNA. This is how the post-transcriptional modification of the mRNA will be happening. And after the post-transcriptional modification, the functional mRNA will be then transported. In case of the eukaryotes, the functional mRNA will be then transported out of the nucleus into the cytosol, where it will be moved on to the ribosome and thereby helping the process of the protein synthesis. Okay, the information in the exon sequence of the RNA will be used for the synthesis of the proteins. Okay. So that is the major function of the mRNA. Now let us look at the ribosomal RNAs or the rRNAs. The ribosomal RNAs, in case of prokaryotes, there are mainly three kinds of ribosomal RNAs are there. 23S rRNA, 16S rRNA, as well as 5S rRNA, okay, which is present in the both the larger subunit as well as in the smaller subunit of the ribosome, you can see. And the most abundant RNAs in the cell are the ribosomal RNAs, okay. The most abundant RNAs are the ribosomal RNAs, and in case of prokaryotes, three major kind of rRNAs are there. That is 23S rRNA, 16S rRNA, as well as 5S rRNA. In case of eukaryotic ribosome, there are mainly four kinds of rRNAs are there. 28S rRNA, 18S rRNA, 5.8S rRNA, as well as 5S rRNA, okay. And they are also distributed between the larger subunit, that is a 40S, that is a 60S, as well as the smaller subunit 40S, okay. The 28S, 5.8S, as well as the 5S rRNAs are present in the 60S, that is the larger subunit of the eukaryotic ribosome, whereas the smaller subunit contains the 18S rRNA. In case of prokaryotes, the larger subunit contains the 23S as well as the 5S rRNA, whereas the smaller subunit contains the 16S rRNA. So in the ribosome, in addition to these RNAs, a group of or abundant proteins are also present. These RNAs together with proteins make up the ribosome composition. So that is what is about the ribosomal RNA. So now let's have a look at the transfer RNA. So the transfer RNA or the tRNAs, they are relatively small RNAs and they consist of a single strand of RNA which is folded into a precise three-dimensional structure. So now let me show you the let me show you the structure of the tRNA. So usually this is actually a two-dimensional two-dimensional structure of the transfer RNA, and this kind of structure of the transfer RNA is called as a clover leaf structure. Okay, the structure of this RNA when drawn in two dimensions, the hydrogen bonding pattern of all tRNAs will form a clover leaf structure. Okay, so this is a typical clover leaf structure of a transfer RNA, which mainly consists of 4 arms are there. Okay, 1, 2, 3, 4. Okay, there are 4 main arms are there in the clover leaf structure of the transfer RNA, and the 2 essential arms which are required for the function of the tRNA are the first one is the amino acid arm. And I told you earlier, the major function of tRNA is to carry the amino acid, okay? Because we know for protein synthesis, amino acids are the backbone, okay? Amino acids has to be added depending on based on the codons in the mRNA, the particular amino acid has to be carried to the ribosome, okay? That function is carried out by the transfer RNA. That is why it has got the name also transfer RNA, which transfers the required amino acid to the ribosome for the protein synthesis. So here you can see on this is the amino acid arm which will be esterified to the particular amino acid. Okay. For example, as per the codon, if a leucine amino acid has to be carried to the ribosome, then here you see CCA, cytosine and adenine. So the adenine residue of this amino acid arm will be esterified with that leucine amino acids, forming a leucine amino acyl tRNA. Okay. Actually, there are 2-3 steps are there involved in the process which is carried out by the amino acid tRNA synthetase enzyme, but I am not going in depth for that. So here you can see the amino acid arm has a major role which is involved in the esterification with the particular amino acid which has to be carried to the ribosome. Okay, another one important arm in the structure of tRNA is the anticodon arm, because this anticodon arm will be interacting with the codons of the mRNA. Then only the information will be information in the codon will be translated or transferred to the anticodon arm of the transfer RNA. Okay, so these 2 arms are very essential for the tRNA. In addition to that, there are another 2 arms are there. One is a D arm. This D arm contains one modified or unusual nucleotide dihydrouridine is present in the D arm. Okay, and the 4th arm of the transfer RNA is a T-Cy arm, which also contains 2 unusual nucleotides. One is a ribothiamidine, another one is a dihydrouridine. Okay, so so T-Cy is a pseudouridine. Okay, T is a ribothiamidine as well as a pseudouridine. These are the 2 unusual nucleotides present in the T-Cy arm of the tRNA. Okay, these are the 4 major arms which are present in the clover leaf structure of the tRNA when you draw it in a 2-dimensional structure. And here you can see the complementary bases will be pairing here together. And some tRNAs will also have an extra arm. A 5th arm also will be there, an extra arm, which will be varying in size, okay, which will not be present in all the tRNAs. Only some tRNAs will have an extra 5th arm will also be there. Okay, so this is how the structure of the tRNA, that is a clover leaf structure, and the function is to carry the required amino acid for the protein synthesis. Okay, so these are the major 3 RNAs which are present in the cell. Okay, now let us classify the RNA based on their functions. Okay, here you can see RNA, as all of you know, the RNA which is involved in the synthesis or coding of the proteins are the mRNAs, okay? Whereas the RNAs which do not have the coding function, they will be coming under the category of the non-protein coding RNAs, okay? So the mRNAs are generally known as the coding RNAs, whereas the tRNA as well as ribosomal RNA is coming under the category of the non-coding RNAs. But in the category of non-coding RNAs, there are several other RNAs are also there, okay? So let's have a look at the various non-coding RNAs. The non-coding RNAs include the housekeeping non-coding RNAs. So as we know, the cell will be having some housekeeping function to maintain the basic functions of the cell and to maintain the homeostasis of the cell, okay? So the housekeeping functions of the cell will be maintained by these non-coding RNAs, which include ribosomal RNA, transfer RNA, small nuclear RNA, as well as small nucleolar RNA, okay? These non-coding RNAs, so earlier it is believed that students, some of the non-coding RNAs are having no functions, but later on research have proved that they also have functions not only in the regulation, even in the housekeeping activity also, okay? So the ribosomal RNA and tRNA, already you know what is the function of these two RNAs, because they are, even though they are not directly coding the proteins, but they also involved in the process of the protein synthesis, because the ribosomal RNA is part of the ribosome where the protein synthesis happens, and the transfer RNA is the one which carries the amino acids required for the protein synthesis, okay? So, and for each cell to maintain its functions, it has to keep on synthesizing the housekeeping functional proteins also. So, in that way, ribosomal RNA as well as tRNA have a major role in the housekeeping activity of the cells, okay? In addition to housekeeping non-coding RNAs, there are several regulatory non-coding RNAs are there, which helps in maintaining the gene expression patterns, okay? So the regulatory non-coding RNAs can be classified based on their length, okay? They are mainly classified into two types, short non-coding RNAs as well as long non-coding RNAs, okay? So the long non-coding RNAs are generally having more than 200 nucleotide length, whereas the short non-coding RNAs are generally having less than 200 nucleotide length. So here you can see a list of the short non-coding RNAs, which include a microRNA, that is miRNA, siRNA, that is small interference RNA, then PB-like RNAs or piRNAs, as well as caRNA. Okay, these are coming under the category of the short non-coding RNAs, whereas those non-coding RNAs, if their length is more than 200 nucleotides, they will be under the category of long non-coding RNAs, which include LANC RNA, circ RNA, eRNA, as well as NAT, etc. Okay, these are some of the examples of the non-coding RNAs. Now, especially these non-coding RNAs, the short non-coding RNAs, miRNA, as well as siRNAs are under research from last 1 decade. The research on miRNAs and siRNAs are still going on, and I don't know how many of you remember this year's even Nobel Prize has been in the field of medicine. Even the Nobel Prize has been awarded for some discovery related to the miRNA. In tomorrow's class, I will be asking you, please refresh your memory and search and tell me what was the role of microRNA in the discovery of the or for the prize winning Nobel Prize for the this current year. Okay, so microRNAs, actually they are emerging as one of the potential area in the research. Okay, so about this non-coding RNAs and their structure prediction, we will be discussing little later. Now mainly we will be focusing on the RNA structure prediction of the coding as well as the tRNA and rRNA. Okay, so now what is the need for the prediction of the RNA secondary structure? Okay, so the importance of RNA secondary structure, we are going to discuss about the importance of RNA secondary structure. Okay, all the participants are there now. Yes, please. Yeah. Will you please tell which are the different RNAs what we have discussed? Nobody is responding. Are you all there?
Coding or RNAs and non-coding rRNA.
Can you tell me the what was the coding RNA we discussed?
mRNA.
mRNA. mRNA is a coding RNA. Okay. So now let us look at the importance of the RNA secondary structure. Okay. Why we have to predict or why we have to look at the secondary structure of the RNA? It has both functional as well as regulatory roles. Okay. So what is the functional role of the RNA secondary structure, which helps to determine the shape and stability of the RNA molecule, and it also creates binding sites for other molecules such as proteins or small RNAs, and also this RNA secondary structure form catalytic sites for the ribosomes, that is the RNA enzymes. Okay, these are the functional role of the RNA secondary structure. So now the regulatory role of the RNA secondary structure. At the gene regulation level, this RNA secondary structure will affect the stability of the mRNA. It also influences the translational efficiency of the RNA. It also controls the RNA processing such as the splicing mechanism. Okay, so the secondary structure of RNA is essential for maintaining the stability of the RNA, and it has both functional as well as the regulatory roles are there. Okay. So the folding of RNA into specific secondary structure is very essential for performing all these functions. And many emerging applications of RNA, including gene silencing, genome editing, and creation of complex nanostructures, they rely on the inverse folding. Actually, many algorithms which predict the RNA secondary structures, it is based on the inverse folding property. Okay, they rely on the inverse folding, the design of RNAs that fold into specified secondary structures in their minimum free energy state. Okay, so what is that? I will explain you in the next 1 or 2 slides. Okay, so the RNA secondary structure, many of the algorithms are relying on the inverse folding of the RNA into the secondary structure with the minimum free energy states. So the mRNA structure prediction, what is the significance of predicting the mRNA structure? The secondary structure of the mRNA helps to control the how the proteins fold. Okay, the secondary structure of mRNA helps control how proteins fold. It is also essential for the gene expression. It affects how genes are expressed, and it is also important for predicting the secondary structure of mRNAs helps to design the RNA based therapeutics. Okay, so the predicting the mRNA structure is very essential for protein folding, for analyzing the gene expression, and also to develop the RNA based therapeutics. Okay, so what is the secondary structure of the RNA? What are the components in the secondary structure of the RNA? Let's have a look. Okay, this is how the secondary structure of the RNA will be looking like. So that it will be having a stem region where the base pairing occurs. Here you can see wherever the complementary matching bases are there, the base pairing occurs within a single RNA molecule that will form the stem region of the RNA. Okay, so wherever the complementary bases can be formed, that will form the stem region, and there will be hairpin region will be also present in the secondary structure of the RNA. That is the point where a RNA chain reverses its directions to allow the base pairing. Okay, so then the secondary structure of RNA, it consists of a stem region is there. There is a hairpin region which helps to reverse the direction of the base pairs to allow the process of the complementary base pairing. Okay, and wherever there is no complementary bases are there, then there will be a small bulges as well as the large loop regions will be formed. Okay, wherever this matching bases or complementary bases are not there, then what will happen that the single bases can bulge, forming a small bulges, or they can form a large loop regions where the bases will be present but there is no complementary base pairing. Okay, and sometimes even RNAs can form pseudonode. Okay, so in this structure pseudonode is not shown. So what is meant by pseudonode means it is the bases involved in a loop, they pair with the other bases outside the loop. Okay, but usually the RNA secondary structure prediction tools are avoiding the pseudonodes. So in the algorithms, they are not involving the pseudonodes. Okay, and here you can see in this structure, in addition to the stem region and hairpin region and small bulge and big loop regions, you can even see in some single stranded regions where there is no complementary base pairing is not there. Okay, so this is how the secondary structure of the RNA looks like, having the various regions. Okay, stem region, hairpin region, small bulge or large loop region, as well as the pseudonodes, and even single stranded regions are there. Okay. And now move on to the RNA structure prediction methods. Okay, there are mainly two RNA structure prediction methods are there based on first one is based on the base pair maximization. In this method, they will be trying to looking at the maximum complementary base pairing. Okay, but what is a disadvantage of this base pair maximization is that such structures are not thermodynamically stable. Okay, so most of the algorithms are avoiding this base pair maximization because this base pair maximization technique, the RNA secondary structure is not stable. Okay, so the second method is by energy minimization, which is the most commonly used method for the RNA secondary structure prediction because it is highly thermodynamically stable. Okay, so the energy, I told you earlier also on one slide that energy minimization word was there. Okay, so with the lowest free energy, okay, the lowest free energy structure will be the most thermodynamically stable structure. Okay, so many of the RNA secondary structure prediction algorithms are using this energy minimization method for predicting the RNA structure. Okay. So now we are moving on to the prediction tools, which are the various RNA structure prediction tools which can be used for predicting the RNA secondary structure. Okay. So here, what I will do that. So here you can see the first one is a RNA fold web server. Okay, the first one is an RNA fold web server. The URL I will post onto the chat box also. You can also try along with me, or after I explain one, then you can try. Okay, so I have posted the link. Here you can see. Now I'm going to open this. Website. Are you able to see my screen?
Yes, ma'am. Manju, ma'am.
Yes, Manju, to sharing.
Okay, ma'am. So the first screen is so here you can see I have come to the RNA fold web server. This is one of the server which can be used to predict the RNA secondary structure. Okay, so here what they have asked first is to paste or type your sequence here. Okay, so first you have to type your sequence here, but the RNA sequences, second, I have to. So all of you know that how to retrieve the gene sequence. I think all of you are very well aware now. How will you retrieve a gene sequence, participants?
From the database.
From the database, okay. In the same way, I'm going to retrieve the RNA sequence also from the NCBI website, okay, National Center for Biotechnology Information. I'm moving on to that site. So, so here I'm going to take the example of the insulin gene, okay. Here I'm taking the gene. Okay, so how to retrieve the mRNA sequence only I'm showing you. So I'm giving the so let us take the Homo sapiens insulin gene. So so I have given the insulin Homo sapiens gene. Okay, so here you can see various, so it is giving a lot of data. So but I require the exact insulin gene. Here you can see I have got that with an gene accession ID 3630. So I am clicking on to the insulin gene. So first you will be getting the gene sequence of the insulin. From there you have to find out the mRNA sequence. Okay, so this is the gene sequence. Now all of you know how to retrieve this gene sequences. But here, as we are going to predict this RNA secondary structure, we should have the RNA sequence. Okay, so you have to scroll the browser to the bottom. There you will be having, here you can see NCBI reference sequences. Okay, rough seq or NCBI, you have to come down. Okay, you have to scroll the cursor so that it will come almost 3/4 of the web page. It has come down. So here you can see NCBI reference sequences. Okay, the RefSeq, where there you can find out the mRNA sequences of the gene. Okay, so here you see down it is given mRNA and proteins. So now I am clicking on to the mRNA sequences. Okay, so from the gene, how to take out the mRNA sequence I have shown you. So here you see Homo sapiens insulin transcript variant 1 mRNA. And they have asked here, you see in the RNA fold web server, they asked for a FASTA format. So I'm giving here the FASTA format. Okay. Then, so I got the FASTA format. Either you can say already moms have explained you how to save the FASTA format, but here I'm copy pasting the FASTA format into the web server here. Okay. So I have pasted the FASTA sequence here, and here you see the what are the algorithms they have used for the RNA folding for the secondary structure, minimum free energy method. Okay, MFE it is called as, and all the parameters I'm keeping as such. The default I'm leaving it, and I'm planning to proceed it. Okay. So what you have to do that first, you have to go to the NCBI or even Galaxy also you can use there, and you have to go to the site, and from that which gene you want to get the mRNA sequence, you have to type it there, and after that, you have to go down to the almost 3/4 of the web page. There you will be getting a rough sequence. There you can find out the mRNA and the protein. There you have to click on to the mRNA sequence. Okay, which will get, from there you have to take the FASTA sequence and paste the FASTA sequence in the RNA fold web server. Okay. So here you can see now the result without taking much time. The result has come. The sequences are here, and here I will show you students. Here you can see how the base pairing is happening. Here you are seeing the brackets, see the brackets which you are showing here, seeing here, it is only towards the one side. So what does it mean by that? There is no complementary or no matching base pairing happening. Whereas you look at here you can see there is the brackets are showing to the opposite sides. Okay, in the opposite way they are showing. So that which shows that there is, so these sequences which are here, they are involved, only these sequences are involved in the process of the complementary base pairing. Okay, so depending on whether your bases are undergoing a base pairing, the notions will be given here. Okay, here you can see it is only towards the one side, and here again you can see it is only towards the one side like that. Okay, now let us have a look at the secondary structure which has been predicted by this RNA fold web server for our insulin mRNA gene. Okay, actually they have given two structures. One is a minimum free energy method using a secondary structure, and another one is a centroid secondary structure. Okay, two structures, very colorful structures they have produced, and the base pairing also you can see here. And now let us have a look, and which one is the most thermodynamically stable structure also let us find out. Okay, so this one is a minimum free energy method. They produce the secondary structure. This is one centroid method. Okay, and again let us go up. Here you can see the free energy of the thermodynamic ensemble is here by the MFE method is minus 201.83 kilocalories per mole. Okay, here the minimum free energy is minus 201.83 kilocalories per mole by the MFE method, and the second one is a centroid secondary structure. Its minimum free energy is minus 150.80 kilocalories per mole. So I told you earlier, the secondary structure prediction is based on the minimum free energy. That structure which has the lowest minimum free energy, that is most thermodynamically stable. So participants, now tell me which one among this MFE or the centroid secondary structure, which will be more thermodynamically stable now? Participants, I have shown you, are you all there? I have shown you two RNA secondary structures of the insulin gene. One is having the minimum free energy is minus 201.83 and another one is a minus 150.80. And I told you the one with the lowest free energy will be having the be highly thermodynamically stable. So tell me now which structure will be more thermodynamically stable now?
First one.
Minus 150.
Minus?
201.
150.
First, let me ask you, minus 150 or minus 201 is a lower value? Yeah, in the chat box, I'm getting some answers.
Minus 201 is lower, ma'am.
Minus 201 is lower. So I told you the thermodynamically stable structure will be the one which has the lowest free energy utilization. Okay. So as per that, we can make sure that the minimum free energy structure, okay, the structure, secondary structure produced by the minimum free energy method with the delta G of minus 201 is the most thermodynamically stable structure. And here also you can see the difference in the structure. Okay. Even though it looks almost similar, here you can see in the centroid structure, the it is almost like the bulges with where the complementary base pairing is not there. They all have come to the center of the structure. Okay, whereas the minimum free energy structure is somewhat most spreaded kind of structure it has produced. Okay, so based on this minimum free energy, so there in this web server they have given two structures. So based on the minimum free energy, so we can conclude that which structure is more thermodynamically stable. Okay, and again down again they have given a graphical representation also for the minimum free energy as well as the centroid also. And now it is very clear to us the minimum free energy structure is the thermodynamically stable with having the lowest free energy of minus 201.83. Okay, so this is how you will be predicting the secondary structure. Okay, and once you get this RNA secondary structure, what you can do that, because that is towards the advanced side of the bioinformatics, then you will be designing some other RNAs which will be able to target the secondary structures. Okay, that already we discussed the functional and regulational role of even for the stability of the RNAs, the secondary structure is essential. Okay, so if you have a secondary structure using some other algorithm tools, you can put this structure there and find out some other or drug molecule which can bind to this secondary structure, or some other non-coding RNAs which can bind to these RNA molecules. All those can be done once you predict this secondary structure. Okay, so this is the one web server which you can use for predicting the secondary structure of the mRNA. Okay, so now let us move on to the next RNA structure prediction tool. The next one is a UNAFold. It is a unified nucleic acid folding and hybridization package. Okay, so it is one of the oldest and widely used tools for the RNA secondary structure prediction. So the key features of this structure include it predicts the most thermodynamically stable structure. It generates alternative suboptimal structures also, and it is one of the user-friendly web interface. Okay, so now let us go on to this UNAFold. So here you can see the UNAFold web server in which RNA folding form is there. Okay, so I'm going to paste the same sequence here. Here also you require the FASTA format, and I think it is already copied only. Anyway, let me once again do that. I'm pasting the RNA, the FASTA sequence, and I hope everyone is clear with how to retrieve the RNA sequence of the mRNA sequence of the genes also. So now I have retrieved the mRNA sequence, the same sequence I am using for another web server. Okay, and all these parameters I am default I am keeping. I am not making any change. So later on when I give the assignment to you, you can do some modifications here and see how the structures are varying. Okay, so now I am giving the option here to fold the RNA. Okay, so let us see how this because the same sequence we have given in another web server. So let us find out how this RNA folding from the UNAFold, how it is going to predict the secondary structure of the insulin mRNA. Okay, so it is taking a little time, and here it is given maximum 2400 bases for an immediate job it will be taken up, otherwise it will be little delayed. Okay, and I hope whatever we have discussed so far, is it clear to everyone? Are you all able to follow, participants?
Yes, ma'am.
Only one is only. So here the result has come for the RNA folding results. Here you can see. Now let us go to the, so in case of this RNA fold web server, So in the unifold, how the result has come here, you see, from structure 1 to almost possible 28 structures have been predicted by using this RNA folding form from the unifold, okay? Whereas in the RNA fold web server, we have got only 2 structures, okay? Whereas in this one, you can see all the possible ways, okay? That is why they have, in the PPT, I have shown you all the suboptimal possible ways, that's how the structures can be predicted. All those they have included in case of this unifold, okay? From almost 28 structures they have provided here. And here you can see the structure 1 here is, okay? The structure 1 here is having a delta G, or the free energy changes here is minus 199.50. In RNA fold observer, what was the lowest free energy? In RNA fold web server, what was the lowest free energy structure? We said that is thermodynamically stable. We said, now, what was that? What was the free energy there?
MFE.
MSC, yeah. And what was its free energy? What was the value there? Free energy value?
They have given in the chat box.
It is already there in the chat box. The number is already there. Yeah, somebody already, it was minus 201 by the MFE method, it was minus 201. So, but here you see, using this unifold, the delta G is minus 199, okay? So, if you do a comparison between the web servers also, you can see the when between this unifold and RNA fold, if you see, RNA fold was having a much more thermodynamically stable structure because its free energy was minus 201. So now let us see the structure which is created by using this unifold method. So they will be providing here, you see, this is how the structure, the first is they have already predicted almost 28 structures for all the possible structures for the insulin mRNA gene. But here you can see the most I have taken the, actually if you go back here, you can see the most stable one among the various structures with the delta G. The most stable is the structure 1 only, having a delta G of minus 199.5. The remaining all are minus 199.2, minus 199.1. From there up to the least delta G is minus 189, okay? So the most stable one is having the lowest delta G of minus 199. So the structure which predicted by this unifold web server is, it is also here, if you see, it is also somewhat similar as the RNA fold web server, having all this loop is there and the stem is there wherever the complementary bases are there. And here you can see some of the bases where the base pairing, because his C to C the base pairing is not possible, and wherever the base pairing is not possible, it is forming a small, here you can see the C is having no complementary base here, so it is forming a small loop here. And this here a large loop you can see where there is a no complementary base pairing is there. Wherever the base pairing is there, it is forming a stem structure, and the stem structure is there, then the loop structure is there. So like that, the the secondary structure of the RNA has been predicted here, okay? And there is some similarity between the structure, because as the sequence is same, even though algorithms, they will be changing slightly here and there. The structure is almost the same, even for the, here you see, here also this kind of loops or stem with the loops are here. In RNA fold web server also, you could see that, okay, whether it is a centroid structure or if it is a MFE structure also, you could see those three stems with the loops were there in the RNA fold web server also, okay? So this is a second web server which you can be used for predicting the RNA secondary structure. And here one more thing is there, participants, here you can see there is one energy dot plot is there, okay? So what is meant by energy dot plot means it is a graphical representation which will tell you how the strong the complementary base pairing, it is coming or not, let me see. So this is how you will be getting the, for the insulin gene, which bases can be involved in the complementary base pairing, and the intensity of that base pairing is denoted by the thickness of the dots also. Okay, some of the bases will be not if the dot thickness is less, that means the complementary base pairing is not possible there. So like that, an energy dot plot also you can be obtained by using the this secondary structure. Okay, so that is what is about the second web server which can be used for the predicting the RNA secondary structure. Okay, now another third one web server which can be used for predicting the RNA secondary structure is its name itself is RNA structure. Okay, so now let us look at the RNA structure. So actually this this is actually a downloadable package. If you can download and use it, otherwise even you can use the online tools also. Okay, so we are going to use the online one. Here the option is there, predict a secondary structure. Okay, so here they have asked again, either you can paste the file or sequence title. So I'm putting the insulin, but here in RNA structure you cannot paste the FASTA sequence. Okay, so what I will do that FASTA sequence cannot be used in the this RNA structure web server. So I have removed the FASTA sequence, only the normal sequence has been added here, and the nucleic acid selected is the RNA here, and all the parameters, the default delta, I am leaving like that only. So later on when you are trying, you can change all these and see how the structure is getting modified. Okay, so now I'm submitting the query. Yes, ma'am.
They are asking for the link for RNA structure tool.
Sure, ma'am. I will paste in the chat box, ma'am. So meanwhile it is loading. Let me paste the link from here. So copy link. I'm pasting the links here. Already one I have already pasted. The second ones also I'm pasting here. So all the three web servers, first I have pasted a little time ahead before I have pasted here already there. That is the RNA fold web server I have already pasted, and now the Unifold as well as the RNA structure web servers also, the link I have pasted here. So you can open that, and instead of insulin you can select your own gene, and from the NCBI, you have to download the RNA sequence, especially if it is an mRNA sequence, because if it is a non-coding RNAs, then another website is there that I will explain you a little later. So after that, you have to paste the sequence there. Here you see, now in the RNA structure web server, we have pasted the insulin sequence. Here also you can see the structure is almost similar as what we have seen earlier, including this stem is there, the loop is there, all the three the stem loop structures are there. But the way how they have positioned is little slightly different than the other web servers. Okay, so here wherever there is no complementary base pairing is there, that single small bulges are there, whereas the large loops are there, and the stem is there wherever the complementary base pairing is there, and here you can see the stem loop as well as the even this is the hairpin structures. Okay, the hairpin structure is also given there. Okay, so this is how you will be getting the secondary structure. So, and here you see this web server is also providing you almost 20 structures for the same gene sequence. So the same mRNA sequence of insulin, it is providing you 20 structures. Okay, so you can go to that each one and see, and where the here also it is and here you see the delta G given for this structure is minus 198.3. Okay, so I can tell you students, we have used the three web servers. One is RNA fold, another one is Unifold, and the third one is RNA structure. Among the three, the RNA fold has given the lowest minimum free energy with the minus 201. Okay, minus 201 having the lowest minimum free energy. So I can say that among the three the structure produced by the MEF, minimum free energy utilization by the RNA fold, is having the lowest energy delta G, whereby that is thermodynamically most stable. Okay, so it is giving a minus 198.3, whereas the RNA unifold was giving a minus 199. Okay, so this is how the RNA secondary structures, especially if you have an mRNA sequence, you can find out the RNA secondary structures for the mRNA sequences. Okay. So this the same thing only. I have just pasted it in the PPT, and I hope it is clear to everyone. Is that clear to everyone, participants? And how to search also?
Yes, ma'am.
So now, just sharing the screen.
Please, students, respond in the chat box at least.
So now let us move on to the non-code. So mRNA, that is a coding sequence, and it's a structure prediction using the various, especially 3 web servers we have discussed. Now let us see the various tools which can be used for the non-coding RNAs. Okay, and as I mentioned earlier, the housekeeping non-coding RNAs are the ribosomal RNA, tRNA, snRNA, snRNA, etc. Okay, now let us first focus on the ribosomal RNA as well as the tRNA. Okay. So the housekeeping non-coding RNAs, ribosomal RNA and tRNA, perform essential roles in the protein translation, which we have already discussed. And what are the non-coding RNA structure prediction tools? Let's have a look. Okay. So the non-coding RNAs, first we are going to look at the ribosomal RNA as well as the tRNA coding tools. Okay. So the non-coding RNA databases, this I will post for you also. I'm copying the link. Especially for the tRNA and rRNA, you can use this link, that is rFAM. Okay, RNA family it is. So let us go to this rFAM. So this is how the rFAM web server looks like. So that is RNA family. The rFAM database is a collection of RNA families, each represented by multiple sequence alignments, consensus secondary structures, and covariance models. Okay, so I'm going to the families. Here you can see the examples, SAM, Homo sapiens, the various species they have given, and I'm going to browse with the families here. Okay, I clicked on the families here. So here you can see the various RNA types, whether it is, and remember here you will not get this mRNA sequence or mRNA structure. You cannot be able to predict here, because especially it is for the non-coding RNAs. Okay, so miRNA, microRNA, snRNA, small nuclear RNA. So like that, various non-coding RNAs, even the CRISPR sequence also given here. Okay, various non-coding RNAs, its structures can be predicted by using, and the various organisms can be predicted by using this rFAM. Okay, so but we need to find out which RNA you have to find out. Okay, for that I'm going, here you see on the left side tab, I'm going to click on the sequence search here. Okay, so here it is asking enter a single RNA or DNA sequence. Okay, so if DNA sequence is there, they will transcribe and they will take up the RNA sequence from that. Okay, so to search this RNA sequence, what I'm going to do here is, here on the top you can see powered by RNA Central. To search the RNA sequence here, I'm going to click this RNA Central here. Okay. So when I click on the RNA Central, so what is RNA Central? That is also another website, the non-coding RNA sequence database. Okay, you can get the non-coding RNA sequence database from the RNA Central. Okay, so here I'm going to browse the sequence of which RNA you want, any tRNA or rRNA. Let us browse. So here you can see text search. I'm going to browse the sequence here. So I clicked on the browse sequences here. So we are done now. We have already done with the mRNA secondary structure prediction. Now we are looking at the non-coding RNA structure prediction. Okay. So here you can see, when you come to this RNA Central, you can see the non-coding RNAs. So among that, various non-coding RNAs you can see. So I am going to take this site. First, let us have a look at the ribosomal RNA. Okay, so I am going to click on the cytosolic 18S ribosomal RNA. So because ribosomal RNA and tRNA are also non-coding RNAs, so to find out the secondary structure of a rRNA or non-coding RNAs, we can use the rFAM, that is RNA families rFAM1 website. In that, to search the sequence, you have to go to the RNA Central, okay? It's taking a little time, this RNA Central. I think I have a PPT also for that. So actually, when you take the sequence, when you click on that in the PPT analysis, so it will be showing you, okay, whether it is a Homo sapiens. If you give the option for Homo sapiens, it will give you the Homo sapiens small subunit ribosomal RNA. Okay, so the various 18S rRNAs are coming. So let me see whether it is come or not. So here it has come. Homo sapiens human RNA 18S ribosomal N2 is there, and exactly Homo sapiens small subunit ribosomal RNA 18S ribosomal RNA is there. Okay, so now we are discussing about the non-coding RNAs, how to retrieve the sequence and the secondary structure of the non-coding RNAs we are discussing here. So the Homo sapiens small subunit ribosomal RNA. So when you click on the cytosolic, let us go back. First, what I have done that I have selected the rFAM. In that I have given the sequence search. In the sequence search, I have gone to the RNA Central where you are getting the rRNA, and I have clicked on the cytosolic 18S rRNA. So the various Homo sapiens, because 18S rRNA is part of the eukaryotic. Okay, so the Homo sapiens rRNAs it is giving. So I'm selecting the Homo sapiens small subunit ribosomal RNA. So then, so here you can see various options it has given. And from this, you can select the 2D structure of this Homo sapiens ribosome. So here you can see the various gene cards, the Homo sapiens small subunit ribosomal RNA, and various gene cards for that. And the gene and the sequences are given here. So let us look at this secondary structure. Let us look at the secondary structure of this Homo sapiens small subunit ribosomal RNA. Okay, so I'm clicking on to the 2D structure. So this is how you will be, this is how you will be getting the secondary structure of the small subunit ribosomal RNA. Okay, so if you want to take out the non-coding RNA, if you zoom it, you will be getting and you can see it will be coming to the colored sequence it is showing. If you zoom further, you can see how the base pairing is happening here. Okay, so you can see a difference between the normal coding RNAs as well as the non-coding RNAs, how it is happening. So here you can see how the colors are formed, and this is how you will be getting the, you can zoom in and zoom out to see the base pairing also. Here also the loop structure is there, as well as the secondary structure, as well as the stem and stem structure is also there, and complementary bases wherever they are pairing also you can see that. In addition to that, here you can see without any pairing single standard structures are also there, and loop structure is there. And here down if you go, you can see based on the sequence whether they are complementary base pairing. If base pairing occurs between the bases, here you can see the brackets are coming as opposite to each other. Wherever the base pairing is not happening, the brackets are only towards one side only. Okay, so which gives the notation that whether the bases are pairing or not also we can identify. Okay, so this is how the I have taken the rRNA sequence using the rFarm, okay? So in the same way, you can even check the tRNA sequence also, okay? So now let us have a look at the tRNA sequence also. So now let's have a look at the tRNA structure, how we can find out. So for tRNA structure, actually, I will show you the PPT here. So tRNA structure also you can find out using the same URL, one that is rFarm, or another one URL is also there. I'll post the URL link in the chat box. And as I mentioned initially itself, students, what is the importance of these secondary structures? Okay, once you predict a secondary structure, then you can find out what are the functional ways, also how the drugs can target to this, whether the drugs can bind to these RNA molecules. So all those properties can be exploited once you have the secondary structure of these RNAs. Okay, so for tRNA two observers, even the rFarm you can use, or even a tRNA tRNA scan is also there. Okay, so let us first try with the rFarm again. So I'm again going to the families here. Again, I'm going to the RNA central here, searching the sequences. So here you see non-coding RNAs, various non-coding RNAs are there. So I'm going to here, you can see the tRNA, okay? And as I mentioned you, for the tRNAs, for each amino acid, a specific tRNA is required for the protein synthesis, okay? So its cells will be having a lot of tRNAs. So I'm going to specifically take one tRNA that is leucyl tRNA, okay? So leucyl tRNA, humans or Homo sapiens tRNA, anticodon arm is there. So let us see any specific tRNA. All these are giving the anticodon arm. Okay, then let's go to the Homo sapiens human tRNA anticodon arm. So for both the rRNA as well as tRNA, we can use this rFarm as well as the RNA central for retrieving the sequence as well as the secondary structure, okay? It's still loading only. And the sequence also, so actually in the rRNA, I forgot to show you the sequence. Here you can see down towards the, if you scroll down, you can see the sequence of the tRNA also, okay? So the leucyl tRNA sequence, you can see that is anticodon arm of the leucyl tRNA. We have selected the RNA, that is Homo sapiens tRNA leucine, it's the anticodon arm, okay? So here you can see the sequence also, you can see the show sequence option is there. And the structure is also on the top, you can find out the structure also. What is the 2D structure of the tRNA also you can see, okay? So there it is gone. So this is the structure of the tRNA using the rFarm as well as the RNA central, okay? So this is also having a stem loop structure is there, and stem loop structure is there, and wherever the complementary base pairing is there, without base pairing the loop is there. So this is how you can predict the secondary structure of the non-coding RNAs also. So now I have shown you both the rRNA as well as the tRNA. Okay. Now let us have a look at the next one another web server which can be used. Especially this is specifically for the tRNA. Okay, I'm opening that one. Sorry. tRNA scan SEO. Okay, that is specifically for the tRNA sequence search and tRNA secondary structure prediction. Okay. So here they have given an option. They have given an example tRNA sequence, or if you want to give specifically select an tRNA sequence, what you can do that. From the RNA sequence, earlier we have seen the RNA sequence. From there, you have to take up the RNA tRNA sequence. So here I am taking one sequence, which they have given here. Just to, I have copied that. And here the FASTA sequence I'm pasting here. We have to give it the sequence name also here. And run tRNA scan. Okay. So this is another one, a web server, which can be used for predicting. This is specifically for tRNA only. So this is how you will be getting the base pairing for the tRNA. What is one's example tRNA sequence which they have given. I have taken that. This is how here also you can see the wherever the complementary base pairing is there, and without pairing also the bases are there in the loop. This is how you will be deriving this structure here. If you see, this structure almost look like the cloverleaf structure of the tRNA, which I have discussed with you earlier while discussing the tRNA structure. Okay. So this particular tRNA structure is almost having that cloverleaf structure only. Okay, where the complementary base pairing is there, the loop is there without base pairs and all. Okay, so this is how the non-coding RNAs, that is especially rRNA and tRNA, can be also secondary structure of this tRNA and rRNA can be predicted. Okay, so that is what is about the RNA structure prediction. Especially in today's session, I have discussed with you the mRNA secondary structure prediction as well as the non-coding RNAs, especially tRNA and rRNA structure prediction. Okay, so hope this was clear to everyone. On the participants, whether the second structure prediction is clear to you. Did you try that? Did anyone of you try with the any link given there?
Yes, ma'am.
Is it working for you?
Yes, ma'am.
So with this, so in tomorrow's session, we will be discussing with the microRNAs and other non-coding RNAs and how their structure can be predicted and what is the significance of their structure prediction. We will be discussing in tomorrow's session. So today, I have discussed with you the RNA structure prediction for the mRNA as well as the tRNA and rRNAs. Okay, and I hope it was clear to all of you. Padma ma'am?
Excuse me, ma'am.
Yes.
I'm having a doubt, ma'am.
Yeah, please tell.
Ma'am, for RNA structure prediction from Matthew's lab, we have to we should not use faster sequence. We should
we should use normal sequence only. Faster sequence it will not take.
Can you show that step once again, ma'am?
Yeah, sure. I will show you. Matthew's step. Let me go to the PPT where it was there. So I think it's there. So this is the one. Okay. So what you have to, oh, sorry, I have gone to the download. You have to use the online tool. Okay. And predict a secondary structure. And what you have to do here that, so here the mRNA sequence of the insulin is here now. I'm copying that, and when I'm pasting here, what I will do that, I have removed all those FASTA format. Okay, only the nucleated sequence. Did you see that?
Yes, ma'am.
So only the nucleotide sequences I have pasted here. And I have selected the RNA here and then submit the query.
Okay, ma'am. Thank
Clear,
you, ma'am.
clear now?
Yes, ma'am.
Anyone else have any doubts? So I request all the participants to please use this online web servers, and I will be giving you an assignment also. So there you have to predict the structure. So I will be giving you some gene names. So you have to predict the secondary structures for that. So please practice yourself for the mRNA as well as the two non-coding RNAs, that is the rRNA, as well as the tRNA structures which we have discussed so far. Okay. Padma ma'am?
Can you explain the tRNA scan once again?
Your voice is not audible. Anusha, can you speak little louder?
Now?
Yeah, now you are audible. Tell.
Ma'am, can you explain the tRNA scan once again?
tRNA scan you want to explain? Yeah, sure. Okay. Let us go to the URL for the tRNA scan. Here it is. So this is a tRNA scan. Here I have taken only an example tRNA sequences which they have given. Okay. So earlier we have taken first sequence. Now I am taking the second sequence, and you have to make sure that the FASTA format it should be. Okay. You have to copy paste the FASTA format only, and again go back and paste it here. Okay. And you should give a name also. Okay. So the same name, whatever they have given, I have taken. So the FASTA format only you have to paste here and run tRNA scan. So then down you can see the download, the structure, how it has come. Predicted tRNA structure, you have to click on to the view option. Okay.
Okay, ma'am, got it.
Got it. Anyone else? Anybody having any doubt, participants? So I hope the structure prediction was clear to everyone. In the upcoming sessions, we will discuss about the more non-coding RNAs, especially the miRNA and siRNA prediction tools also we will be discussing. Okay, Padma ma'am?
Yeah, yes, Manju. Yes, Manju. Okay, so I hope all of you have followed the prediction of RNA structure and the significance of predicting the RNA structure. Can anyone just tell me why you have done all this exercise so far? Quickly, what was the idea of predicting so many forms or so many tools we are using? Outcome of your understanding today, spending so much to learn things?
Yes, participants, please respond.
What is the final take-home message you got from today's class?
You can type in the chat box, participants. Please type in the chat box. Whatever you have heard. Yeah, I could find some answer. Others, please. If you're not able to. Yeah, one has come out. Others. Hope you're all near your systems. We will see who will be the next participant typing the message. What are the different types of RNA, which is coding? Okay, Srinidhi has given some answer. Okay, that's nice. Others? List out all the RNAs. Okay, yes. Okay, using different tools. Yeah, for drug binding sites. Interesting point. That's it. Okay, non-coding function. Fine. Yes, UG students, you can list out all the RNAs, what you have heard till now. Yeah, exactly. Okay. Only one has responded. I have, excluding all the faculty, I can find 40 participants and I get response only from few. To determine the shape and stability of the molecule.
Among all these RNAs, what you have studied today, which you find is more stable, which you find is more interesting in structure-wise? Did you find any RNA structure interesting?
Okay. tRNA.
You found
Why
tRNA
tRNA?
structure?
Why tRNA? mRNA. Krishdev has Krishna Veni, I think so. You have responded as mRNA.
Nice student, nice participants. I think you are really enjoying the course. Are you enjoying it? Doing these exercises? Using
capital Y.
different types of tools?
I expect capital Y now. Okay. Is it, I said why you are putting Y and there is no N? Is it true? Because I said why it is Y, or is it, it's true? Okay, good. That's what we want. Thank you for the response.
So you have seen the sessions in the past 4 or 5 days. You have seen the DNA structure, extracting the gene sequences, and then we have gone through identifying the coding regions, exons, introns, overlapping regions, and then doing amplifying the gene. So from this understanding, and today you have been explained and taught about or trained to use RNA. So I'll give 5 minutes time to all of you. Can you quickly create a small project for 2 minutes on interrelating all these topics what we have discussed? I will give you some time, 5 minutes I will give to all the participants. Can you devise a small quick project relating all these 5 days of training what you have undergone? So the next set of classes which you will be going on from today, next day onwards, it would be follow-up of what you could understand till today. So you have done genomics, you have done transcriptomics, and you will be doing a little more of transcriptomics. So now can you quickly make some sense out of the whole thing by creating a project in 2-3 minutes? How you would like to use it further? Yes? I'll give you 5 minutes time, then we'll go further. Yes or no? Otherwise, we have to do something else now. Yes? Any response from students? You want to do it? Or are you doing it?
See, what we are expecting is not a very big project, right? You have to retrieve a sequence. Instead of taking one, take a list of sequence. We have read clustol. Then you do the multiple sequence alignment. Take that one gene from that one. Find the overlapping genes. Or if you can do some PCR analysis, mean in silico PCR, you see whether you can do. And whatever we have taught, You do step by step some of the topics correlating and try 1 or 2 things and show it to us. That is what we want. We are not asking a big exercise to be done. Okay. Today also ma'am was telling about RNA, right? So you take the sequence, put in the tool, give the output.
What is the DNA structure for the gene sequence? What is the RNA structure for that particular gene? How it is getting transcribed to RNA? What could be the structure for that particular mRNA? Whichever is of your interest.
So time is now 7:53. So by 2000, whoever has finished any one activity, what we have given now, you can share the screen and tell to us. So we are available here so that we will know that when we are teaching, when you are doing, you'll be able to do. But in our absence also, you should be able to do it. So you have to experiment by yourself. So without any help from us. So if you get stuck anywhere, if you now you can clarify it immediately, you can proceed at any time in our absence also, right? It's learning process. So please try something with whatever you have dealt in all these 5 days and try to share with us. You can share the screen later and you can show it to us. So we will wait. We are giving you some time. Please try to do it. Hope everyone are working. Anyone has tried? Or do you want some time? It's 8.
I'm like anyone, or we have to do all?
What you have done till now?
I did multiple sequencing.
Only one you have
Multiple
done?
sequence alignment. Like I'm doing the other ones.
Do that one. We will wait for some more time. Anybody else has finished any other task? Like anything two? Padma ma'am?
Yes, ma'am.
Padma ma'am?
Padma ma'am.
Launa? Because participants are quietly working, I think. So next 2 minutes, I thought I'll ask them to share the screen till whatever they have finished. Ma'am, if she says okay, then we will start to check. So anyone has finished? So till wherever you have finished, maybe you can start to share your screen and show what you have done till now. Anyone, if you're halfway through also, it doesn't matter.
Ma'am, am I audible?
Yes, Srinidhi.
Ma'am, I have explored 3 tools. Should I share my screen?
Yeah, please.
Okay. Ma'am, is my window sharing?
Yeah, yes.
Okay, so I have taken 2 sequences from NCBI, which
Okay.
is first one is Homo sapiens ka insulin, and the other one is insulin of this organism. I've performed cluster for this, and I got the results like this, ma'am.
Okay. But
And
why
then
the alignment is not in, you have taken how many sequence, ma?
only 2, ma'am.
Cluster omega, if you want to perform, first thing remember, you have to take multiple sequences, more than 3.
Okay, ma'am.
Yeah, always you should do. That's why you're not able to get your result. It's not comparing and giving the conserved or whatever region. So remember point number 1, when you do cluster omega, you have to take more number of sequences. Okay. Second thing, when you shared the screen, first one you have taken correctly, insulin you have taken, right?
Yes, ma'am.
Homo sapiens. But the other one, what you have taken, what is that one you have taken?
This is also insulin 1.
Insulin 1.
Yes, ma'am.
What is the size, number of bases? How many bases are there in the second one you have taken? This one you're telling?
Yes,
What
ma'am.
is the size?
Just a minute, ma'am.
Here organism is different, ma'am. Vanta ma'am.
That's what. See, it's not wrong, but still, you have to take thing is more than 3 sequences. Second thing, insulin when you're taking, you try to take different organism, and here I don't find the size also is not related. I think you're showing a big size for this one.
Okay, so the sequence size also has to be similar, ma'am.
Not 100% similar, but if you take 1000 and if you compare it with 5000, how you will get the similarity or dissimilarity regions, right? So if you take 1000, C for 900 or 800, or C for 1100 or 1200, like that you try to take and compare.
Okay, ma'am. I'll try to do this.
Yeah, sure, please. What is another thing you have tried?
This was GenScan. I did with the human.
Okay. So you have tried with GenScan. Okay. And what is the third tool you have tried?
PCR, primer tree.
Oh, okay. For which gene?
Yes, ma'am.
Which gene you?
Insulin. Humans, Homo sapiens.
Okay. Good work. But first one, whatever you have done now, you try to do it with more number of sequence, so that after my class you had phylogenetic analysis, right? So there ma'am was insisting, telling us that you have to take more number of the sequences so that the output will have a better results. Okay.
Sure, I'll do that.
Please try that one. Thank you so much.
But here, 1 minute, you haven't taken the same one. In the previous GenScan, you showed different sequence. Here, you are showing a different sequence.
No, you are not. Sequence, ma'am. Homo sapiens insulin sequence.
In the previous one, you showed some carpenter and some sequence.
For the phylogenicity, I took two, from the 2 organisms, right? So I took from Homo sapiens
She
as
took
well.
a different one.
Okay, so no, why when you are working, you have to use the tools on a same set, then you could follow it up properly, because here one, there one means when you are doing a work, it would be better that you pick up something and follow it up with whether it is working with all the tools what you have learned. Every tool will work with a different sequence, but what is it that you will understand for one particular sequence, you will understand better when you apply different tools on the same sequence. If you are facing an issue there, then see, suppose you have taken this, then you want to design a primer. Now, did you take a primer blast or did you do any other sequence? That's why I'm saying you are showing chromosome 11 here FASTA, and output is for chromosome 11, whole chromosome you have taken, chromosome 11. So what is there in that, whether there is an exon or an intron, what is there? We are not sure, right? So when you are looking, when we say that you should do some small quick work, then you should plan in such a way that you pick up something where you can retrieve the sequence and do the steps, follow the steps, design a primer, do a PCR, do an alignment. So you have to follow a particular sequence. The thought behind asking you to do is how well you could design some quick analysis. That is what I was looking at.
Sure, ma'am. I'll do that.
Yes.
Thank you so much, ma'am.
Yeah, thank you, Srini. Thank you. Prakriti has done MSA.
Yes, ma'am. I did MSA of these proteins, ma'am. Albumin from rat, human, and Canis familiaris, dog.
Okay.
So that I did, and I also made I also performed like phylogenetic tree.
Okay.
Those are the two
Yeah,
things I did.
then maybe you can share it so that Padma ma'am can give some inputs.
Yes, ma'am. Is it visible?
Yeah, we are
able to see. Yes, ma'am. So I went to Uniprot. I got the sequences, 3 sequences, and I performed the multiple sequence alignment.
Here you have taken all the mammalian sequences.
Yes, ma'am.
You should.
I
It
found.
is going to be a conserved sequence.
Yes, ma'am.
So many
These
stars
are the
are
results.
there.
Yes, ma'am.
That is.
And this was the phylogenetic tree.
Okay.
This was the radial version.
Okay.
That's it.
Okay.
I'll take other sequences too, ma'am, from
please.
non-mammalian animals.
Sure.
Again.
I could find Manognia. Galaxy you have used, right? Prakriti can stop sharing. Yeah, thank you. Manognia, can you share?
Yes, ma'am, I will.
Others next? Anyone is there who has worked with a continuous way like how Padma ma'am is asking you? Okay, you've done.
I did it on chromosome 22, ma'am.
Same thing. Okay.
As I got the result.
Same result you got.
Yes.
Okay, nice. Only one thing you did, right? Now?
Yes, ma'am. Actually, I did catch up with you. But I did not keep them like that. So I just removed the browser and all.
Okay.
Yeah, so for now, this is there.
Okay.
So I got the same result as you.
Okay, that's nice.
Thank you.
You check your workflow also from the history. You have the 3 lines, right? And see that. You can save it and keep also. In case if you want in future, you can go back to that and create workflow. You click, you will get edit. So there you can Okay, thank you.
Yes,
Anybody
ma'am.
else? Anybody else? Yes, yes, I can see the hands being raised. Please, what you have worked with?
Ma'am?
Mahesh?
Ma'am, in the gene prediction, RNA gene prediction, can we use 2 sequences, ma'am, at once?
You can use, but you can use an RNA fold or RNA server. There is an option where you can compare 2 sequences. That part we have not shown today, but there is a Manju is here. Manju is there?
Yes, yes, ma'am.
Manju?
I think Manju is not to be seen.
Okay, anyway, so you can, Mahesh, in the RNA server, there is an option where you can RNA fold or RNA web server. In one of them, there is an option where you can do comparison of 2 sequences. You can have that option of comparing the RNA sequences. But today we have not shown that to you. We have just shown with only 1 sequence.
Okay, ma'am.
Yes. Anyone else?
Am I audible, ma'am?
Yes.
Ma'am, so I've done this RNA fold one, ma'am. Like just now we had the class, right?
Yes, yes. You completed?
Yeah, yes, ma'am. Yes.
Okay.
Is the screen sharing, ma'am?
Yes, it is. You are sharing the screen.
Ma'am, so I took the research article of the Nobel laureates, ma'am, like this year's Nobel winners.
Okay.
So I took the mutant microRNA gene mutant which they worked on to find the function. So I went to this hyperlink and then here I scrolled down and selected the whole sequence, like the whole genome sequence of the mutant microRNA.
Okay.
Then I went to the RNA fold web server, ma'am. Then I gave the input sequence and then I got the structures here.
Padma, ma'am?
Yeah.
You are saying no, ma'am?
Yeah.
Please చూడండి ఒకసారి.
So ma'am, here in MFE, I'm getting a lower score, I mean lower value.
Okay.
So it is more thermodynamically stable.
Yeah.
So here is the structure.
So how is this centroid structure different from this structure? Did you put it in 2 different forms, RNA structures? Yeah, you can see the difference between the MFE structure and the centroid structure. MFE structure is little spread out and easy to look at, whereas the centroid structure is a little closed and it has become very compact compared to the MFE structure. So obviously, because it is coming close together, the free energy associated there is slightly higher when compared to the relaxed form, that is the MFE form. So that way you could infer the MFE form is having better, MFE is a preferred secondary structure compared to the centroid structure. So the other fold also, other server also is giving you the same expanded structure for that is the MFE structure only you are getting. You can see the comparison of structure between these 2. Yes. So centroid is always showing some constraint on the structure.
Padma, ma'am? Now, this participant has taken mutant, right? So now he can take the native one, that is which is not mutated, and if he runs, they compare and see this with that one and try to tell what is the structure difference or what it is.
This is only predicting the structure, so this is not based on a mutation or anything. It is only giving the structure. So if we take the sequences like we do alignment, only there we can predict what are the differences or SNPs or something like that.
My idea is, in case if he takes the wild type and does it,
Will
free
the
energy
energy be
structure,
different? Will the
that he can do the comparison. No, I'm
Yeah,
telling.
for that we have to show the link where that comparison of sequences has to be done. So we need to.
Ma'am, but the gene itself is a lethal gene, ma'am, which I took.
Acha.
Okay.
Okay,
That's it,
ma'am.
ma'am. Thank you, ma'am.
Okay. Thank you. Yes.
Okay, students, then participants, I have a small information to share with you. I'm just sharing the screen. I have already put the list on your WhatsApp group. The same screen I'm sharing also right now. Just give me a minute. Is the screen visible?
Yes, ma'am.
So, not this one, no, this is not the one. Another one, sorry. I put the wrong one. And just, you have it on your screen, then you can download that. You have it on your system, Vanita? Hello?
Ma'am, I have it.
Yeah,
Shall I
I
share
shared
it?
the wrong one, I think. Yeah.
Yeah, I'll share to them.
Oh.
Already they have in the WhatsApp group.
Yeah,
They must
WhatsApp
have
group, at
seen.
least for us to explain it also.
Okay, then I'll share it. This is the list. Ma'am, it's visible, right?
Yeah.
You can see the list. So 3 participants are highlighted in red. You have given the transaction ID, but the supporting document as a proof what you have uploaded, in that one we are not able to get your transaction ID, because maybe when you're taking a screenshot, there will be some ad coming, right? You have taken that screenshot and it is hiding the transaction ID. So you 3 people have to post personally, either to Padma ma'am or to me, your payment details, which is clearly showing your transaction ID. Okay. And we have at present, from the registration data, we are able to get 50 participants' names and the transaction ID. In case if anybody has paid and your name is not been reflected, you can please approach Padma ma'am or me, because you have our numbers in the WhatsApp group, right? And you can show the payment details, and we will and try to do next what we are supposed to do. So the same list is posted in your WhatsApp group. Okay, na Padma ma'am? Shall I stop sharing? Padma ma'am? Okay, so if anyone is having
If anybody's
issue,
name is not there, they will just see and let us know. Just keep it for 2 minutes. Just now I got a photo. Who says this? Who sent me? Varalakshmi. Varalakshmi is not there in this list. Varalakshmi, are you in the class?
Yes, ma'am. UPI ID is not visible. That's why I send the ma'am.
Okay. Your name is there, no, in this list? Okay. Okay. Anyone else? All other names are there and participants whose names are there are in the class. Today only 40 people are there, 10 people are missing.
Ma'am, my name is not there.
You please share the screenshot. Anusha is not there here. You did not share your transaction details to us. You did not submit the form with the transaction ID.
Anusha is not there, ma'am.
But she has sent the transaction details. This one, when did you pay, Anusha?
Now on 20th.
20th you have paid. So you didn't submit the form.
I submitted the form.
Then the transaction ID you didn't give. Why it is not reflected here then?
I've entered it wrong or something.
Okay. Anyone else? Did you send the money to account or did you? How did you do that? Did you do GPay?
I did GPay to the account details that were mentioned on the form.
Okay. So anyway, now your details are there. Anyone else? Okay then, students. I hope you.
Ma'am,
Yeah.
am I audible? Ma'am, my name is not there.
Your name?
Insha.
Insha. You send your payment details.
Yes, ma'am. I've just sent it to you again.
Now I got only two. I got you must have sent to Vanita ma'am. Which number did you send? 00013 or any other number?
Ma'am, 13.
Yeah, 013, right?
Yes, ma'am. Just a minute.
I
I'll
didn't get
resend it.
I didn't get it. You did not submit the form.
Yes, ma'am. I had already submitted that day.
What is your name?
Insha.
Huh?
Insha. INSHA.
No, that name I didn't find in the registration, this thing.
Even another one, another name she told just now. Anusha. Not
Anusha.
Anusha, Insha. They too sent now.
Okay.
Okay, students. So if anybody else, you can just look at the list. It's already there in the WhatsApp group. You can check and get back to us. I hope you had a good learning experience today. Kamala, you can wind off the session.
Yes, ma'am. Yes, ma'am. Students, Vanita ma'am will post the attendance link in the chat box. Okay, please fill that attendance, today's attendance. Are you able to access the attendance link? Vanita ma'am just posted here. Everyone able to access this link, ma'am? Attendance, today's attendance list link.
Just respond. Participants say
Yes,
yes.
ma'am. We are able to access it, ma'am.
Yes, ma'am.
Okay.
Once you completed, please put a message.
Kamala ma'am, out of thanks చెప్పండి.
Yes, ma'am. So today I just want to thank to Dr. S. Padma ma'am. Thank you, ma'am. Dr. Manju Devi ma'am for this today's speaker. And Vanita ma'am. Thank you, ma'am. Shall I wind up?
Okay, I'll just stop recording 1 minute. Others,
Excuse
if
me,
you have
ma'am.
finished, yeah?
Ma'am, can you just read